Thank you very much for this enlightening presentation. We have the discussion after the three presentations now, and that's why we move immediately to the next question of cancer. And as you see, this conference is designed to make a bridge from fundamental research to application. This involves an industrial stage at some point, and that's why we are very happy to hear about the unresolved problems in cancer from a person from industry, from Novartis, Dr. Jens Würthner, who will share us about the challenges in front of us when we want to treat cancer in novel ways.
Well, thank you very much for the invitation. I started in oncology as a clinician, and then 10 years ago or so made a switch to the industry, and I'm involved in drug development now, and I need to remind myself not to say that to American Border Police when they ask me what I do. They just don't like the word drug, so it's pharmaceutical development of oncology products, if you will. So this is my disclaimer and disclosure. And so this is just a high view picture on oncology these days. I think we've made tremendous progress over the last decades in terms of therapy, but also in terms of prevention and diagnosis, and that is shown on the left side of this slide. Now, on the right side, you see the problems that we face. The population is aging, and so the incidence of cancer is actually rising. It's several million people this year, approaching 13 million, but this number will actually increase to over 20 in the next few years. So I was asked to talk about the challenges in oncology, and I want to do exactly that. I don't want to tell you how nanomedicine can work. That's something I hope you will tell us. So I really want to focus on an unbiased view of where I think the challenges lie. So I tried to subgroup them into two big categories: the scientific and clinical challenges and the drug development challenges. And obviously there's some degree of overlap, and you'll see what I mean when I talk through the slides. So in terms of the scientific and clinical challenges, you all know about primary resistance. I don't want to get too deep into that. Just maybe a few words. There are a certain number of tumors which are just inherently difficult to treat. So pancreas tumors are an example. Glioblastoma, tumors of the nervous system, are very difficult to treat, and patients very quickly to succumb to the disease. Melanoma for the longest time was one of the diseases, and fortunately there has been some great progress made over the last few years where for decades there were no new approvals in that disease area, and now there are actually drugs coming to the market that are very effective, and I will show you one of those in just a minute. I want to focus on secondary or acquired resistance, which is what happens when patients are treated and then acquire resistance to the therapy and progress with their cancer. I want to talk a bit about toxicity, and then also the problems or issues we face when we want to combine our drugs. How do we best combine them, and how do we actually then schedule them? That's still quite an unresolved problem. So let's talk about secondary resistance, and for the sake of the argument, just let's think Basel is a tumor cell. And so we have an important signal that's coming in through the SBB railway station that needs to be delivered here to the Congress Center. And what we can do with drugs nowadays, we can inhibit such a signal, and we can kind of create a roadblock. And this is a big boulder here on the Wettstein-Brücke, which could theoretically prevent the tram going to the Congress Center. So what tumor cells then do is they find pathways around this. They just find alternate ways to deliver the signal to the cell nucleus and turn on the cell to divide. And these, you've probably seen many of these diagrams. They all look very theoretical, and so I want to show you what this really means. So this is a publication that came out a few years ago in the Journal of Clinical Oncology from colleagues of Nick Weigel. And what you see on the left side is a patient with a metastasized malignant melanoma. And that patient was then treated with the first BRAF inhibitor that was then subsequently licensed as a drug. And you see this is almost a miraculous response that this patient underwent. And what the people, the researchers did, they took biopsies from the metastasized tumor on the left side, and they sequenced a number of genes, and I just highlighted a few here where they showed some mutations, and the one at the top is what we call the driver mutation. That is actually the mutation that made these cancer cells divide, the BRAF V600E mutation. And against that specific mutation, a small molecule inhibitor was developed, and that led to this response. Now, unfortunately, after the patient had been on that therapy for a couple of, few months, this happened. And so the cancer came back. And so Nick and his team, they again sequenced, they took again a biopsy and sequenced it. And what they again could very nicely show, you need to read the paper to get the exact detail, but they could show that a new driver mutation occurred in an enzyme called MEK, which is downstream of BRAF, and that then led to these melanoma cells again to grow. And so you could say, okay, well, this is kind of a, it looks terrible, but the concept seems simple. You have a patient, you sequence the tumor, the patient relapses, you take a biopsy, you sequence again, and hopefully by that time you have a new inhibitor in the pipeline, and you just give that new inhibitor, and you keep on carry on doing this. Unfortunately, it's not quite as easy as this for two reasons. One is, in this patient, all the tumor lesions are very, they are dermal, and you can very easily biopsy them. As you've seen before, the patient with the non-Hodgkin's lymphoma, the lesion was in the thorax. So very often, actually, you have to do CT-guided or ultrasound-guided biopsies, and it's sometimes difficult to get a biopsy from patients because very close to the tumor might be important vessels or nerve structures that you might harm. So it's not always easy to get biopsies, in fact. And the second problem that was not addressed in this publication but in subsequent publications is that actually different metastases develop different driver mutations. So it's not just all these metastases are driven by this MEK mutation, most likely, but different, even within the same tumor. There was a publication focusing on renal cell cancer, and they looked at the big primary tumor, and they took samples from different parts of that tumor, and they could show different driver mutations in different parts of the tumor. So it's probably not done just by adding one additional drug, but you probably have to have an armamentarium, and to decide individually on each patient which combination is right is obviously a big challenge. So this is just another cartoon to highlight that there are other mechanisms that the cell can evade these roadblocks. So, for example, they can just push the boulder into the Rhine in this case. So that means they just push the drugs outside. And I've seen some posters already which address these transporter proteins that just pump the drugs that are harmful to the cancer cell. They just pump them outside. So that's also an issue that needs to be addressed. So this is just the overview of what I've just talked through, some of the mechanisms. The third point here is the immunotherapy. That's the latest kid on the block, if you will, and has shown some remarkable efficacy in melanoma again, and other tumor indications are now coming closely behind where immunotherapy might work. And again, cancers develop mechanisms to evade immunotherapy, and that's still an evolving science. Some of the molecules that drive this immune interaction are being downregulated by the cancers. Some cytokines that are known to be immunosuppressive are secreted, and again, these need to be addressed in the future. So let's talk about toxicity. Does anybody know what this toxicity is? I hope we haven't reached it yet. It's the PowerPoint poisoning. So I think I'm more than halfway through, and we shouldn't really reach this, I hope. So toxicities of targeted and immunotherapy agents. We all know that chemotherapies are toxic, and but it's not that targeted therapies are not toxic. They have different kinds of toxicities that we also need to consider. So if we want to talk about, usually we divide them into on-target and off-target toxicities. So the on-target toxicities are theoretically the ones you can predict that are linked inherently to the inhibition or stimulation of your target. So in terms of certain antibody therapies, you might have heard that there are toxicities like cardiotoxicity for HER3 antibodies and skin toxicities for EGFR and so forth. So these really affect targets in normal cells, not in the cancer cell. But you can also, if you overdo it, so to speak, in your target cell, you can also have toxicity there. So if you treat tumors too hard, you can get something which is called a tumor lysis syndrome, which can basically clog up your kidneys by all the debris that you generate when you lyse these tumor cells. Or here in this picture, some of you might remember the T. Gennaro accident or incident a few years ago when an agonistic molecule was developed that stimulated T cells, and they just got the math wrong, and they didn't, or the number of receptors on these cells, and so they got an overstimulation of the immune system that led to severe symptoms. And this was a healthy volunteer here in that picture who, had to go to the intensive care unit and suffered from necrosis at hands and feet. So these are all on-target effects, and you can also have these off-target toxicities where you hit other targets, and people use the word iatrogenic effects and so forth, where sometimes or often it's not quite known what actually the problem is. And then you can also have allergic reactions when you use immune therapies and antibodies. The next topic I wanted to just highlight is the issue of optimal combinations. So many of the big companies have dozens of compounds in their pipeline, and we are faced with the question, how do we best combine them? What really makes sense? And sometimes you can make a lot of hypotheses. Let's look at the cell cycle. Should we do mitosis inhibitor first and G1 phase inhibitor? You can make a lot of arguments theoretically, but in the end you really need to test it out. And you need to test it not just in cell culture and in animals, but ultimately the human system is the one where you need to prove the point. And that's very costly and time intensive. And I don't think we have found a very good way to address this. Obviously, we're trying to, do these large-scale experiments, but again, they are in vitro, and the translation to the human system is not always straightforward. One example that comes to my mind, a few years ago, I was working on HDAC inhibitors, and for another company, and I thought these molecules are very interesting. They do a lot of things. They turn on a lot of genes. Unfortunately, you always go into the clinic with a single agent molecule. You just give one therapy, and the efficacy was just not overwhelming and rather disappointing, and the whole field kind of lost interest. And still, you see publications coming out addressing that this could be very powerful combination agents, but now also the patent life, which is, as I've shown here, you usually do your patent early before you start your clinical trials, and then it's after 20 years they have patent expiry. There are some ways to extend this a bit, but once you get into the clinic, you only have 10 to 12 years usually left of patent life, and when you then find that the compounds are not doing what they're supposed to, and then you start to think about combinations, you just run out of time, and that's a bit of a challenge as well. In terms of drug development challenges, I don't want to go too much into attrition rates. You've all heard the story that too much is falling by the wayside and the costs of development are still too high. Costs of the therapies nowadays are really becoming very high, and I've heard the term financial toxicity, which I found interesting, and we need to, as an industry, address this. There are operational challenges that we maybe can skip here. And I just want to finalize the talk by just looking at the co-development of companion diagnostics and the integration of novel formulations, which may be closer to your heart. So in terms of the diagnostics, what we really try to do these days is find the safe responders. We try to deselect for poor responders or even those responders that might be harmed by the therapy. So when you go back, you need to ask yourself, can we define biomarkers that predict efficacy for nanomedicines that really show the value that you add to certain subpopulations of patients. And the challenge is that nowadays, when you license a drug with a new diagnostic, you need to do this at the same time. So when you come to the end of your confirmatory clinical trials, you have a concomitant submission of your drug application and your diagnostic application. And in the drug label, you then often find this drug is licensed for, let's say, ALK-positive lung cancer patients by an FDA-approved ALK diagnostic. And that's the challenge. You're dealing with two different divisions of FDA, and it's similar or going to be similar in Europe. And you need to marry these two development pathways, and that's not an easy thing to do, but this is where the field is clearly moving. And finally, just an example from my recent experience where we had two different formulations, an oral formulation and an IV formulation. And the IV formulation turned out to be a bit of a liquid soap, in fact, and there were some tolerability issues and regulatory concerns. So people said, well, why don't we develop a nanomedicine for this purpose that was going to be better tolerable. And unfortunately, the numbers that were on the table where this would cost another year of development, and it's going to cost over $1 million. So what happened here was that this was cancelled. We had an oral formulation that seemed to be good enough, and we kind of cancelled the whole nanomedicine part, which is unfortunate because there were some aspects that could have been beneficial. And so I want to close by just suggesting to come up with some kind of generic vehicles that people can just plug their drugs into without then having to really reinvent the wheel every time and add cost and timelines. So with that, I want to close. I talked about the scientific and clinical challenges. We talked a bit about resistance, about toxicity, and how we can the issues about combining new novel therapies. And in terms of drug development, we highlighted now the co-development of companion diagnostics and the integration of new formulations. And with that, I want to close. Thank you.